Students, today we will study about biomolecules. In our previous video, we have studied about carbohydrates and nucleic acids. Today we will start from proteins. Proteins are the heteropolymers containing amino acids, or these are the polypeptide chains. So the types of proteins depending on the amino acid, the number of amino acid residue and the sequence of amino acid. What about the structure of proteins? The proteins exist in four structural forms. Very first is a primary structure. Primary structure is a linear chain of polypeptide molecules. As you are able to see in this, we have the one end we have the amino terminal, amine group is present there, and on the other hand we have the carboxylic group. So this is a polypeptide chain in which the amino acids are joined together by the peptide bond, by the help of peptide bond. The linkage is peptide linkage. Secondary structure. The secondary structure is a result of hydrogen bond. It exists in two forms. One is alpha helix and second is beta plated sheet structure. Now we have the quaternary structure. The tertiary structure is a one structure. Tertiary is there. The tertiary structure in which two or more chains are highly coiled or supercoiling is there. But when we have more than two chains, like here we are able to see the four polypeptide chains when overlapped over each other, that is said to be the quaternary structure. Okay, and the example of this quaternary structure is hemoglobin molecule. Now next we have the classification of proteins. The proteins are of two kinds, fibrous protein and globular protein. The polypeptides, which are arranged in the form of parallel bundles like silk, fibers, keratin, collagen, are the example of fibrous protein. And the globular protein include albumin and globulin, which are coiled and folded. Next we have the simple and conjugate proteins. The simple proteins are composed of amino acids like histone and albumin, and the conjugate proteins consist of peptide chain and the cofactors. Metabolism. Now in this chapter we have two important terms, that is anabolism and catabolism. These terms are related with the organic molecules. When complex organic molecules are formed from simpler ones, like proteins are formed from amino acids, and we have the polysaccharide molecules are formed from simple sugars, these are termed as anabolism. The process is anabolism. The catabolism is a process of broken down of the complex molecule into the smaller substances. For example, the digestion of protein by the peptide. Now we have the primary and the secondary metabolites. The primary metabolites have identifiable functions. These include the carbohydrate, proteins, fats, and the secondary metabolites, the product of certain primary metabolite pathways. The primary metabolites are amino acid, nucleic acids, nucleotide bases, proteins, nucleic acids, and the secondary metabolites include pigments, like we have anthocyanin pigment, anthocyanin carotenoids. Drugs include curcumin, alkaloid includes morphine, essential oil, lemongrass oil, lectin, cocanevaline A, terpenoids, monoterpenes, toxins, aberrant resin, polymeric compounds, rubber, etc., gums, etc. Now, what are lipids? Lipids are the heterogeneous group of organic compound which are formed from fatty acid and glycerol molecule. They are water insoluble and soluble in non-polar organic solvents. Now, there are two kind of lipids, straight chain and fused hydrocarbon with the long hydrocarbon chain. Example, cholesterol molecule. That is a complex molecule. So, there are two kind of lipids, simple and compound. The simple includes oil, fats and waxes, and the compound includes phospholipids, glycolipids and sphingolipids. The simple lipids include the molecule, the simple lipid, we are having the structure of glycerol molecule here, that is formed by the esterification of, the fats are formed by the esterification of glycerol with fatty acid. These include monosaccharide, when one fatty acid molecule is there, monoglyceride, diglyceride and triglyceride molecule is here. Now fats have high melting point and remain solid at room temperature. Now you are able to see the structure of a fatty acid here, that is palmitic acid, and the formula is CH3 CH2 14 COOH. It includes 16 carbon atoms. Now we have the enzymes. The what are enzymes? Enzymes are basically the biocatalysts, or these are the protein molecules. So the characteristics of enzymes are they play very important role in reactions, various processes, biochemical reactions. Enzyme catalyze the reaction of a substrate, and they doesn't enter in the reaction. They works at a specific temperature and pH, and they accelerate the reaction. They speed up the rate of the reaction. We have certain similarities between enzymes and inorganic catalysts. The catalysts remain unchanged at the end of the reaction, and they can be used again. They are released. They are required in very less quantity as compared to substrate. They do not initiate a reaction, but the reaction they lower down the activation energy for the substrate to be converted into a product. It doesn't alter the equation of a equilibrium of a reversible reaction and form the short-lived complexes. Now here we have certain examples between enzymes and inorganic catalyst. The enzymes are protein molecules and the inorganic catalyst are small and simple molecules. Enzymes catalyze a specific reaction, they catalyze any kind of reaction. Enzyme action can be regulated by specific molecules. It cannot be regulated by any other molecule. Now nomenclature of enzyme will add suffix ase to the substrate on which they can act. Example sucrase, protease. To physiological activity, it catalyze the various classes. We have oxidases, dehydrogenases, decarboxylases, reductases. Now we have the classification of enzymes. The very first is the oxidoreductases, which will catalyze oxidation reduction reaction of a substrate. Second is transferases. They catalyze the transfer of the specific group from one substrate to another substrate. Example is glutamate pyruvate transaminase. Third class is hydrolases. They catalyze the breakdown of larger molecule into the smaller molecule by the addition of a water molecule. Liases catalyze the cleavage of specific covalent bond and removal of certain groups. Isomerases catalyze the rearrangement of molecules or atoms within a molecule to form the isomerase. Ligases, as the name suggests, they help in making a bond. They catalyze the covalent bonding between the two substrate to form a large molecule involving the utilization of energy. Now here you are able to see the lock and key hypothesis. We have the substrate here that will be fitted into the enzyme and it will form enzyme substrate complex. Next is a catalytic cycle. The substrate binds to the active site of the enzyme and it will form the enzyme substrate complex. The active site of enzyme is now in close proximity with the substrate and breaks down the chemical bond, and enzyme releases a product of in the end, and the enzyme is again free to take up another molecule of substrate. As you are able to see in this figure, we have maltose. That is a substrate. When it binds to the enzyme, upon enzyme substrate formation, it will release two product in the form of glucose and glucose, two molecules of glucose. Now we have certain factors which will affect the enzyme action. These are temperature. At a particular optimum temperature, the enzyme activity will be maximum. After this, it will be decreased. So at a particular point at a optimum pH, the effect of the concentration, the enzyme activity will be maximum. So with increase in a substrate concentration, the rate of reaction increases, and after that it will become stable. Effects of chemicals. When binding of a chemical reduces, shuts off the enzyme activity, and the chemicals which inhibits the enzyme functioning are known as inhibitors. There are two type of inhibitors. One are competitive and other one are non-competitive. Competitive resembles the substrate in the functioning. Now we have cofactors. The cofactors are the known protein molecule, and the apoenzyme is a protein molecule. Collectively, they will form holoenzyme. That's all.